Welcome to Dr. B.R. Ambedkar Open University. Today topic is Algae General Account and Classification. It is present in UG first year, first semester and PG first year course 1. It will be useful to academic students and competitive aspirants. Now the topic contents are introduction, habitat, thallus organization, cell structure, reproduction, life cycle and classification. Just give me the brief introduction about this topic. What is algae? There are so many definitions for algae. The simple definition is algae are autotrophic, thallophytic, non-embryophytic, non-vascular, spore producing simple plants. Autotrophic means they can prepare their own food material by photosynthesis, and thallophytic means thallus like plant. We can see our surroundings, so many plants, they can identified as a root system, stem and leaf, whereas in algae we cannot identify root system and stem and leaf. They are all together and appear as a club mass, greeny masses appearance, that is called as a thallus, and it is non-embryophytic means embryo does not form in their life cycle, and vascular system also does not form, and finally the end of the life cycle which produces spores. These spores developed into thallus again. This is thallus structure and algae definition. Study of algae called as phycology or algology. The first algae word introduced by Linnaeus, father of algae F.A. Fritsch, father of Indian algae M.O.P. Iyengar. It is first prokaryotic algae and which produces oxygen in atmosphere, because already told you they are autotrophic. Auto means self, trophic means nutrition. So they can prepare their own food material by photosynthesis. That's why they are the first photosynthetic plant and first oxygen releasing plants, example cyanobacteria, cyanophyceae, algae. Now in topic general account. Student, you should remember general account of algae, not only algae, any general account of fungi, general account of bryophyta, general account of pteridophyta, general account of gymnosperms. These are all very important. If you know about general account, you can easily identify about type study. For example, in our syllabus, algae general character 1 unit. Next are all type studies. There, for example, in our syllabus, chlorella, chlamydomonas, volvox, edogonium, ectocarpus, polycyponia, they are all present. They are all will be easy to you if you learn about general account. Whereas in fungal general account, type studies are mucor, rhizopus, puccinia and neurospora. In bryophyta, marchantia, anthoceras and funaria, they are all type studies. And pteridophyta, lycopodium, equisetum, marsilia. In gymnosperms, Pinus, Netum and Welwitschia, they are all type studies. So, before entering to the type studies, we should know about general account. The first, in general account or in any type study, first we can simplify the topic. First, where is algae? Second, how is algae? Third, formation of algae. Where is algae means algae natural place, that is called habitat. How is algae means structure of algae, it is thallus structure and cell structure. And formation of algae, the algae formation by reproduction. So, these three aspects very important in general character or any type study. First, let us see about habitat. Habitat means natural living place, that is called habitat. Here we can see in slide, these are universal occurrence, general habitat of algae, water. So, these are called aquatic. Apart from that, few are terrestrial and aerophytic and cryophytic, epiphytic, parasitic, symbiotic also. And let us see in aquatic, freshwater algae, Oedogonium and Chara, and some marine water, Ectocarpus, Sargassum. And terrestrial, the terrestrial grows on moisture soil. This example, Vacheria and Botredium. Aerophytes, they can present in air. Example, Trentifolia. So, these aerophytes get the water from the air. And cryophytes grows on snow. Example, Encyclonema or Chlamydomonas. Encyclonema called as ice bloom. Next, thermophytes. Thermo means heat. These are observed in hot spring and high temperature. Example, Oscillatoria brevis. Lithophytic. Litho means rock. They can grow on rocks. Example, Ectocarpus or Oedogonium. And epiphytic. Epi means upon. Phytic means plants. They can develop on plant surface. Example, Chaetonema, Rivullaria. Mainly these Rivullaria appeared on angiospermic plant Scirpus. And Chaetophora grow on Neelambo leaf. Next, epizoic. Epi means outside, zoic means animal. Example, Cladophora. The Cladophora grow on snail shell. And endozoic. Endo means inside the animal. Example, Zoochlorella, which is observed in Hydra. And parasitic. Parasitic means, actually before I told you these are autotrophic, that means they can prepare their own food material, but very few shows parasitic nature and symbiotic nature. The parasitic means they can grow on other host and causes diseases, cephaloron, which cause rust disease in coffee plants. And symbiotic nature, in symbiosis, examples lichen and coralloid roots formation, and some of the algae, especially nostoc, grows on Anthoceras thallus. So, this is thallus, this is algae habitat. After habitat, next one is thallus organization. Already told you what is thallus. Undifferentiated roots, shoot and leaf called as a thallus. In thallus, from primitive to advanced one, they are evolved as a unicell to multicell. Unicell, next multicell, in multicell synobial or colonial, next filamentous and parenchymatous. Let us see one by one. In unicellular means the entire thallus made up of single cell, that is why it is called as a unicell. In unicell again two types, motile unicell and non-motile. So, in motile, with help of flagella, they can move from one place to another place. Example, Chlamydomonas. And non-motile, these flagella are absent. Example, Chlorella. Synobium. So, in first unicellular made up of single cell, the entire thallus made up of single cell. But in synobium, these are all synobium. So, group of cell all together form as a group, as a colony. So, the each and every cell attached with mucilage sheath, that is why appeared as a colony, but does not form tissue system, tissue organization, but appeared as a multi-cell organism. This is called as a cenobium, and again cenobium also two, motile and non-motile. The motile cenobium are colonial, example Volvox and Pediastrum. Non-motile hydrodictyon, and it is called as a water net. Next, filamentous thallus. So, thallus appeared as a filament as a unique manner. Before that it is irregularly appeared, but the unique appearance in filamentous thallus, and again in filamentous divided into unbranched, pseudobranched and branched. First one, unbranched. So, thallus, the cells developed one above the another, one cell, second cell, third cell and fourth cell. So, this is the filament, this is also filament, there is no branches. So, this type of thallus called as unbranched. Example Edogonium, Spirogyra, Nostoc, Oscillatoria, they are all unbranched thallus. And pseudo-branched, pseudo means false, not true. We can observe in this slide, it appear look like a branched, but not true branch. This is one filament, this is one filament, they are merely attached with help of mucilage layer. That is why it is appeared as a branched. So, it is called as pseudo branched, example Cytonema. And branched one, we can observe here side clearly, this is filament, this is also filament. So, it is much branched, example Cladophora. And in branched one, heterotrichous also present. In heterotrichous means two type of systems, one is prostate system and another one is erect system. This type of system called as heterotrichous thallus, example Cara and Ectocarpus polycyponia. This is about thallus. And next one, parenchymatous. In evolution process, after filamentous and well developed thallus are formed, that is parenchymatous. It is completely made up of parenchyma cells, and here also completely made up of parenchyma cells. It is most advanced one, laminaria. That means here look like a mini plant. We can observe look like a mini plant. So, halva one example and laminaria another example. So, this is thallus organization. So, if we observe thallus organization, first single cell, next multi-cell irregular and regular manner filamentous and parenchyma. So, this is evolution of thallus. Next, after thallus, any thallus either unicell or multicell, in multicell cenobial, filamentous, parenchymatous, any cell made up of cell. So, let us know about cell structure. Cell main components, cell envelope and protoplast. Cell envelope, again two types, sheath and cell wall. Here sheath only present in Cyanophyceae or blue-green algae bacteria, blue-green algae Cyanophyceae bacteria, and this sheath made up of pectic substances, and the cell wall made up of cellulose. The cellulose is main chemical component in algal member. Even though some of the algae, these cellulose absent, cellulose chemical absent, they are blue-green algae cell wall and diatom cell wall and Caulerpa cell wall. These three cell wall does not made up of cellulose chemical. And next one, so actually this cell envelope act as a wall, compound wall, that means which gives the protection to the cell. Protoplast, protoplast outer layer made up of cell membrane. Next cell membrane, the total portion called as protoplasm. In protoplasm, nucleus and cytoplasm with cell organelles are present. At the nucleus, two types, prokaryotic nucleates and eukaryote nucleates. The prokaryote means nucleus does not form well developed. That means nucleus, nuclear membrane absent, nucleolus also absent, only genetic material present in cytoplasm. Whereas eukaryote, clear nuclear membrane and nucleolus, the both are well developed. Only prokaryotic algae cyanophyceae members, rest of the all chlorophyceae, phaeophyceae, rhodophyceae, all algae eukaryotes. And cytoplasm with cell organelles. Cell organelles are Golgi complex, mitochondria, endoplasmic reticulum, lysozyme, ribozyme, vacuole, they are all. But as it is autotrophic, most important cell organelle are chloroplast. So, chloroplast appeared in different forms. Actually, in higher plants, these are biconcave structure, but in algal member, it is different forms. These are discoid, star shaped, ribbon shaped, reticulate. So, different forms of chloroplast are present in algae. And inside the chloroplast, thylakoids present. In thylakoids, pigments are present. These pigments are chlorophylls and carotenoids, which are classified into xanthophylls and carotins. Let us see first the chlorophylls. The chlorophyll types are in algal member, it consists chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D and chlorophyll E. Chlorophyll A is the universal pigment which is present in any autotrophic organism, whether it is algae or bryophyte or higher plants, anyone chlorophyll A is present, and chlorophyll B present only in chlorophyceae members, and chlorophyll C present in xanthophyceae and phaeophyceae apart from the chlorophyll A, and chlorophyll D present in rhodophyceae member, and chlorophyll E present in xanthophyceae members. And carotenoids, these are xanthophylls and carotins. Xanthophylls are fucoxanthin, which are present in phaeophyceae, basilarophyceae and chrysophyceae, and another type of xanthophyll, taraxanthin, which is present in rhodophyceae members of algae, and myxoxanthin, which is present in cyanophyceae members of algae. Carotins, the mainly beta carotene and E carotene present in algal member, and phycobilins, these axillary pigments only present in cyanophyceae members and rhodophyceae members. These are two types, phycocyanin, phycoerythrin. Mainly phycocyanin present in cyanophyceae members, phycoerythrin which is present in rhodophyceae member. And storage food material, main storage food material starch, apart from that in cyanophyceae members myxophycean starch and floridian starch in rhodophyceae member, laminarin, mannitol present in phaeophyceae member, and in xanthophyceae storage food material oils and fats. So, this is about cell structure. Next, reproduction. What is reproduction? Reproduce, that means one organism produces another organism. This is called as a reproduction. It is carried out by three methods in algal member. One is vegetative reproduction, second one is asexual reproduction, third one is sexual reproduction. So, any method they can produce new thallus. First, vegetative. In vegetative, cell or any vegetative part can develop into new thallus, that is vegetative. And in asexual reproduction, spores are formed. These spores develop from the protoplast, and the spores after formation released out and can develop into new individual thallus without fusion. Without fusion, they can directly produce the new thallus, that is asexual reproduction. And sexual reproduction, in sexual reproduction like formation spores, instead of here formation of gametes, the male gamete, female gametes, the both gametes develop from the protoplast. After formation, male gamete and female gamete, they are fused by fertilization, as a result formation of zygote which is developed into new thallus. This is sexual reproduction. Let us see first vegetative. In vegetative, unicellular structure and multicellular structure. In unicellular, it is carried out by cell division. Here we can see this is one cell, unicell, and is divided into two daughter cell. Before that, first In unicell, nucleus divided into two nuclei, which follows cytoplasm division or protoplasm division. After formation protoplasm, it is separated into two individual cells. So, this is cell division. Example, chlorella. And another one is fragmentation method. It is mostly appeared in multicellular organism. In multicellular organism, this is filament due to any reason by aquatic animals or water tides. If it is break into pieces, each and every piece can grow as a new individual thallus. So, this is fragmentation method. And another one is actinids. These both are formed in favorable condition. In unfavorable condition, inside the cell, protoplasts round up and it is synthesized by number of layer around it. One layer, second, third, like this number of layer formed, and this it is appeared as a thick structure. This is called as a actinid. When favorable condition reoccurred, the actinids are grow as a new individual thallus. This is actinid structure, and it is also mainly found in Spirogyra or Edogonium or Nostoc oscillatoria. And another type of reproduction is asexual reproduction. So, simple in asexual reproduction, first of all this asexual reproduction takes place by formation of spores. These spores may be haplospores or zoospores. So, any type of spores formed inside the sporangium. This sporangium developed from the vegetative thallus. Already explained about the thallus structure. So, thallus made up of cell, any one of the vegetative cell developed into sporangium mother cell. Later it is developed into sporangium and which produces the spores by protoplasm. So, these are the zoospores. Anyway, after formation this type of zoospores, aplonospore difference. Here zoospores with flagella, aplonospores without flagella. Any type of spores, whether zoospore or aplonospore, after released out, it is again developed into new individual thallus. This is asexual reproduction. And another asexual reproduction method is autocolony formation. Autocolony formation mainly occurred in coenobial cells. In coenobial, already it is made up of number of cell. Any one of the cell developed as a sporangium cell, and inside the cell nucleus redivide and forms a number of nuclei, and which is finally developed into spores, and these are finally developed into Volvox colony or daughter colony. Next one, sexual reproduction. Already told you the sexual reproduction takes place by formation of gametes. These gametes developed inside the gametangium. In gametangium protoplasm metamorphose into gametes. The both gametangium, whether male or female, develop from the vegetative thallus, cell. Anyway, after formation of male and female gamete, these both are fused. As a result, formation of zygote, diploid zygote, because if the male gamete haploid, female gamete haploid. That is why the diploid zygote developed. After formation diploid zygote, which is undergoes to meiotic division and produces the haploid spores, and these haploid spore can develop into individual thallus. And this sexual reproduction, three methods. One is isogamy, second anisogamy, third oogamy. In isogamy, as I said, both gametes are formed, male gamete and female gamete. Male gamete developed inside the male gametangium, female gamete developed inside the female gametangium. After formation, this male and female gamete, in structurally these are similar, male gamete as similar as female gamete. So, this type of fusion called as iso. Iso means similar, gamy means fusion. At the male gamete, female gamete, both are similar in structure, both are fused. In anisogamy, one is male one small and female one large. So, this type of fusion called as anisogamy. In oogamy, male gamete small and released out, swim in water, but oogamous, but it is female gamete not released out. It is situated inside the hugonium or female gametangium. So, for suppose this white color is the female gamete, it is not released out, it is situated inside the hugonium. Now, only the released male gamete attach fertilized with female gamete. As a result, by any method, isogamy method, anisogamy method, hugamy method, finally produces the diploid zygote. After formation diploid zygote, now it is undergoes to meiotic division and produces haploid spore, and this haploid spore can develop into individual thallus. Next, life cycle: haploid life cycle, diploid life cycle and isomorphic life cycle. First, let us see haploid life cycle. In haploid life cycle, gametophytic thallus, already told you thallus structure, and which produces the sex organs, male gametangium and female gametangium. From the male gametangium produces male gamete, from female gametangium female gamete. After fusion, after fertilization, diploid zygote formed. Diploid zygote by meiotic division produce haploid spores, and again these haploid spores germinated into haploid thallus. This is haploid life cycle. Diploid life cycle, but the main thallus diploid 2x. In haploid life cycle x, but here 2x. So, from the diploid thallus, male gametangium and female gametangium developed like a haploid life cycle, but here the male gametangium diploid and female gametangium diploid. That is why before formation gametes, before formation of male and female gamete, it undergoes to meiotic division. As a result, here the haploid gametes are formed. Now, these both haploid gametes, male and female, fertilized, as a result formation of diploid zygote, and this zygote directly produces the spores by mitotic division. By mitotic division, here the difference between meiotic and mitotic: in mitotic the chromosome number neither decrease nor increase, but in meiotic number chromosome number decreased off. Here we can see this thallus diploid, gametangium diploid, diploid after meiosis it becomes haploid, meiosis division. But in mitosis, from zygote these spores develop from the mitosis, but mitosis only diploid spores are formed. Isomorphic life cycle: iso means similar, similar morphology, life cycle, diplohaplontic, it means two ploidy, one is diploid, another one is haploid, but these both are identical, identical thallus. First let us see diploid thallus. In diploid thallus, this is sporophytic thallus, sporophytic. From the sporophytic, sporangia developed, from sporangia zoospores are released. These released zoospores developed into haploid gametophytic thallus. Here before formation gamete, before formation of zoospores, it undergoes to the meiotic division. As a result, it formation haploid spores. These haploid spore developed into haploid gametophytic thallus. After formation gametophytic thallus, on the gametophytic thallus, male gametangium developed, female gametangium developed. From the male gametangium, male gametes are developed, from the female, female gametes are developed. Both male and female gametes fertilized, as a result formation of diploid zygote, and this diploid zygote developed into again sporophytic thallus. So, first start with sporophytic thallus and again form the sporophytic thallus. So, this is isomorphic life cycle. And final classification: the first time Algae classification done by Linnaeus, later Wacher classified systematic classification. Next, Eichler classified the algae with fungi, including fungi, and the most authentic classification of algae done by Fritsch. Fritz's main criteria are pigments and flagella and reserve food material, main criteria for his classification. He classified the algae 11 classes: chlorophyceae, xanthophyceae, chrysophyceae, bacillorophyceae, cryptophyceae, dinophyceae, chloromonidae, euglenophyceae, phaeophyceae, rhodophyceae, cyanophyceae, 11 classes. First, the chlorophyceae, the main pigments. Already told you these are the criteria for classification: pigments, flagella, reserve food material. For chlorophyceae, main pigments chlorophyll A and B, carotenoids, xanthophylls. In flagella, 1 or 2 or 4 or more equal flagella present, they are an anterior side, and these both Whiplash and tinsel type of flagella present, and reserve food material is starch. In Xanthophyce, chlorophyll A and chlorophyll B, but chlorophyll B rarely found. Apart from that, chlorophyll E also present in Xanthophyce members, and carotenoids, xanthophylls also present. And two unequal, in Chlorophyce equal flagella and Xanthophyce unequal flagella present, they are anterior side, and tinsel and Whiplash type. Reserve food material, fats and leucosin. And Chrysophyce, main pigments chlorophyll A and B, carotenoids. Flagella number 1 or 2, unequal or equal. Anterior may be lateral sides also formed, both Whiplash and tinsel type. Reserve food material, oils and leucosin. Bacillariophyce, chlorophyll A and C, carotenoids, one anterior flagella present, and reserve food material, leucosin and fats. Cryptophyce, chlorophyll A and C, carotenoids and xanthophylls, unequal flagella at anterior side. Reserve food material, starch. Dinophyce, chlorophyll A and C present, and carotenoid xanthophylls also present, and two unequal flagella, Whiplash type, and they are arranged at lateral side. Reserve food material, starch and oils. And Chloromonidae, chlorophyll A and B, carotenoids and xanthophylls, two equal flagella present, and reserve food material, oil. Euglenophyce, chlorophyll A and B, and one or two anterior tinsel type of flagella present, the reserve food material, fats and paramelon. And Phaeophyce, chlorophyll A and C and xanthophylls, and two unequal Whiplash and tinsel flagella are present. Reserve food material, laminar in starch and fats. Rhodophyce, chlorophyll A and phycoerythrin. Here flagella are absent. Reserve food material, Floridian starch. Cyanophyce, chlorophyll A and carotenoids, and C phycocyanin, allophycocyanin. Here also flagella absent. Reserve food material, Cyanophycean starch. Thank you one and all.